Okay.
So we're going to be presenting on TIP60 hat, which is a model for memory deficiency and Alzheimer's disease model. So why TIP60 hat and why Alzheimer's disease? Alzheimer's disease is an irreversible progressive neurodegenerative disease that eventually causes people who are affected by it to be unable to use most basic things. The problem with this is that researchers aren't sure exactly what is the cause of this, and therapy for it is it's the sixth leading cause of death in the United States. So in previous research, they found that disruption of epigenetic gene control mechanisms in the brain causes significant cognitive impairment, like memory loss, and thus is the leading hallmark of Alzheimer's disease. Specifically, histone acetylation has been one of the best characterized of these epigenetic mechanisms that is critical for regulating learning and memory-associated gene expression. So then, why TIP60 hat? So TIP60 hat just means TIP60 histone acetyltransferase, which is basically what's doing the histone acetylation. It shows a high homology in humans, up to 99%. And when I say that, I mean the Drosophila version of TIP60 hat is very close to the human, to our version of TIP60 hat. In previous research, it was found that TIP60 hat regulates the expression of many important learning genes that are associated with memory. It was also shown that TIP60 hat, when it's knocked out in an Alzheimer's disease model fly, causes the downregulation of many important learning genes, and that by itself causes memory deficit.
So that experiment, the downregulation of TIP60, was that done in the fly or the human?
In the fly.
Or was it found in fly? No, yeah, go ahead.
Yeah, this is all done in the fly.
So based on what Sophia told us about Alzheimer's TIP60 hat, we know that when TIP60 hat is knocked out, a bunch of memory-related genes are downregulated, specifically rutabaga gene, foraging gene, and testiculin 2. And we want to determine which of these downregulated genes causes the memory deficits in this Alzheimer's disease model. Any questions? Yes?
How close is the Alzheimer's Drosophila model to the human model? Like, how are you telling what to say about it?
Well, we know that TIP60 in flies, it shares a 99% homology with the TIP60 in humans, and I think it's around the same percentage homology for the three genes.
The phenotypes expressed in the Alzheimer's disease model fly are very similar to the effects Alzheimer's disease has on.
So you look for learning in the fly?
Yeah.
Look for learning? I'm just curious about the Alzheimer's model.
We're looking for like
learning.
memory loss and learning deficiency in the fly.
Like what we're using, well, I'll explain it later, but we're using a courtship assay to test for learning versus not learning.
We'll get to that.
Yeah. Okay. So the experimental part of our project has three pieces. We are going to have a genetic piece in which we're going to knock out TIP60 hat using the GAL4-UAS system and then reintroduce the three specific learning genes with the Lexay-op system. Sophia's going to explain that more later. And then we're going to use immunofluorescence to validate our selective expression. And then our behavioral part of our experiment is going to be a courtship assay, where I'll explain that later. Like, basically, just tests for memory deficits.
So the genetic manipulation part, we're basically going to be using the GAL4-UAS system to knock out TIP60 hat. And at the same time, we'll be using the Lexay-op system to selectively reintroduce the three learning genes that we had mentioned before. And these three learning genes we know are regulated by TIP60 hat. So the importance of using the GAL4-UAS system and the Lexay-op system is that it allows us to specifically target certain areas in the fly. So let's say like a certain area of the fly's brain, like the mushroom body, and express whatever protein we want there. So it allows for like really specific expression.
How?
How? Okay. So we're basically, to have this happen, to have the fly that's going to be expressing the genes we want, have knocked out the genes we want, we're going to have a lot of different strains of flies, and we're going to have to cross them. So the way Gal4 works is that you have a fly, the driver fly, and it has a Gal4 on its genome, and that is right before a tissue-specific protein. So let's say this is the protein encoded for, like, I don't know, something in the brain.
Oh,
So
that's what I wanted to know.
just this.
Yeah.
Yeah. So when this is transcribed, translated, made, Gal4 itself is transcribed and it's made into protein, and the protein Gal4 comes over to the UAS region on the genome, and then that activates the UAS to begin transcribing and translating the effector gene, which is basically the gene that we want. So we would have like TIP60 hat in here, or one of the learning genes in there. But problem is that there aren't the Gal4 and the UAS little regions. They're not exactly in one fly, so we'd have to do a cross of a fly with the Gal4 and then a cross of the fly with the UAS and the protein. So it's going to be a lot of crossing to get the transgenic fly that we want.
I'm sorry, why they're not in the same fly?
I mean, they can. It kind of depends on the
type of fly that we
the
can
type of
order.
fly that we're going to order. So there might be a fly with the Gal4 UAS system all in it, but if there's not, we're going to have to do this crossing.
And then you have to somehow select a fly that has both genes.
Yeah, there's actually a really big stock center called Bloomington, which has like thousands of different strains of flies. So all of them are there, and you can go through and find the one you want, select it and order it, and then you can do your specific crossing to get what you want.
The crossing gives you
It gives you options to like play around with it. Really.
genes in the same fly.
So are you saying you want to create a knockout of TIP60, or it already exists?
That already exists.
Okay.
Yeah, so that's there. Like we want to do this specifically just so we can validate those results and get the same phenotypes that previous researchers have already done, right? And then we're going to be using the Lexay-Lexay-Op system to reintroduce our lineage genes. That system is basically the same, works the same way that the Gal4 system.
How do you select for those flies that have both systems in them? Selection must be hard.
We would have a fly with the Lexay-Op system with one of these 3 genes, and then we'd have to cross it with a Gal4 fly that has the knocked out TIP60 hat.
And
But then you have to select.
But what would the outcome be? Like, what would the fly, how would you determine if flies have both? I think that's what you're asking.
Well, we're gonna
put a an identifying phenotype gene, something that causes an identifying phenotype onto each system to show that it's being expressed. For example, like the example that we used when we were learning about it was we would attach a Curlio gene to the Gal4 UAS system, and we would know that the Gal4 UAS system is present when the Curlio gene is also expressed. So it would cause a
How can you tell if that's expressed?
there would be a
Because it would
phenotype in the fly that we know translate to the Curlio.
So visually.
Yeah,
Curlio would cause a curled wing versus a normal straight.
Yeah. So.
1 minute.
Okay.
So I mean, we've been.
Yeah, I think we'll
we've
give
been
you a little bit of
asking
extra
questions,
time,
So we'll
so.
have extra time,
Okay.
but
Yeah.
just so you know.
Okay, sorry. Selective expression part, we're going to be using immunohistochemistry basically just to validate that our genetic manipulation has worked. Immunohistochemistry is under the umbrella of immunofluorescence, so that's basically just an important application of antibodies to determine the distribution of an antigen in a specific area. So basically you'll have your antibody that will chemically correspond with a fluorescent dye, so let's say like GFP or something. And then those antibodies will bind to your antigen, your protein of choice, and then you'll be able to look under a fluorescent microscope and see where your antigen is located. This is just an example of some of the stuff we hope to see.
So So this is, you're dissecting the brain, out of brains and looking for, and then fixing, doing some sort of fixing on slides.
Yeah.
Do you guys have the anti-TIP60 or
No, we'd have to.
we'd have to buy it or
But
do
it's
you?
available
Yes.
commercially?
Well, that's in our little budget.
Okay.
But
So our behavioral assay is going to be a courtship model. Basically, what happens is after we genetically manipulate our flies, we are going to take a male fly and
with the manipulation.
a male fly with the manipulation and put it with a female fly that has recently been mated. And flies that have recently been mated do not want to mate again for a while. So as this male fly tries to court the female fly, he will constantly get rejected. And then we're going to take this basically trained fly and put it with a virgin female fly and see how long it takes and how often it attempts to court with this new virgin fly. And basically, if his behavior matches a wild type fly, then he hasn't learned anything. And if he's slower, if he doesn't court her as often, then he has learned from the rejection of the previous mated fly. Does that make sense? Any questions?
Okay, keep going.
So we are going to we're going to put the male and female flies together in a chamber and either watch it live or watch it via a video camera that we've recorded. And some of the behavior we would look for is some of the courtship behavior would be the male orienting himself towards the female and following her around, tapping the female with her leg, which is a version of tasting, and vibrating his wing, which is basically him singing. And also if he licks the female out and curls the abdomen and attempts to copulate. We might do we might try and identify all of them, but I think we're going to pick one or two phenotypes to follow. Any questions? So our current team consists of Sophia and I, and Dr. Kirkhart is our mentor, and we usually meet Monday CPs, though sometimes because of scheduling that doesn't always work out. We currently know nothing about flies, but Dr. Kirkhart is going to train us.
Clearly not true.
Dr. Kirkhart is going to train us in all of the experimental stuff we need to learn, and while she's training us, we're going to be preparing for the experiments. So as soon as we're finished with our training, we can start right away.
So is this basically a rescue experiment, right? Is that basically what I'm understanding? So you're going to take you're knocking out TIP60, and then you expect the 3 genes to be, I can't remember what their names are.
Yeah.
3 genes to be affected, and then you're reintroducing those and hoping to rescue behavior.
We're hoping to rescue behavior. What we're
And trying to figure out is which of those genes actually rescues memory or causes the memory.
Why did you select those 3 genes?
Because we know that it's, sorry, go ahead.
Sorry, I forgot to mention that. These 3 genes have previously been researched a lot, and they're shown to be important in different aspects of memory themselves.
And we
Then-
know that they're regulated by TIP60.
Then why are you doing it?
What do you-
If you know they're regulated by TIP60,
But we don't know which one causes,
Is like the main gene that
Which-
disrupts the process of memory when it's downregulated.
yeah,
So that's what we're looking for. Because when TIP60 is knocked out, these genes they don't go away. They're like extremely downregulated, like 3-fold.
Mhm.
So we're trying to figure out which one of these genes is causing the disruption.
So you're saying they haven't looked at those 3 genes individually. That's what you're going to do?
Yes.
No, I mean, I'm not trying to trip you up.
No, yeah, no.
So like,
we're just trying to see when the genes are downregulated, which one actually causes the most memory deficits
By itself,
by itself.
because no one's looked at those 3 genes by themselves before.
No, I think,
In this situation, no.
So how are you going to reintroduce? You're going to reintroduce. You're going to make different constructs for each of the 3.
yeah, I mean, because I think what we're doing is first we're going to focus on the rutabaga gene, because that has I think it was the first memory gene like discovered in
So-
Drosophila.
So you're gonna rescue the other. You're gonna bring back the other two genes.
No. We're gonna knock out all three. Well, not knock out, but downregulate them all
Right.
by knocking out TIP60. And then we're going to, using the GAL4US system,
Right.
and then we're going to rescue only rutabaga using the Lexi-Lexi.
And see what the phenotypes are. See if the memory has returned to that wild type.
But then aren't you?
But you're losing the other two. So I guess the question is, do they all work together, you think? Do you need all three genes present, or I guess, I don't know.
I mean,
It's a related
I
question,
think
I
that's
guess.
what we're trying to figure out. If it, like how it relates to a normal wild type TIP60 and the wild type regulatory genes.
we're not confined to doing to expressing just one. We can try to express two, or try to express all three.
So that's
Yes.
possible
There are
in
many
your system?
combinations.
Oh,
There's-
yeah.
Yes.
Yeah, there are a lot of different.
Because I think there are other systems that are like the the GAL4 and the Lexa
That allows us
systems,
to do
so
that.
we can.
So those are
isolate each-
individually inducible.
Yeah.
Yes.
Oh, okay.
They're
Thanks.
not-
All right. That's what. I was-
But then can you have two, can you choose 2 out of 3 to reintroduce, 2 out of 3?
We would just have each of them on a different system.
So yes, the answer to what you're
Yes.
saying, it sounds like it's yes.
Okay. I'm good.
I guess you can have 2 gal systems.
Oh, no, we'd be using the separate, that's why we thought GAL4 for
If
knocking
you're-
out TIP60, Lexite for breathing.
No, Kirkart.
Sorry.
It's fine. It's fine.
They're very close to correct.
I just out of
The
curiosity.
answer is still yes. The
You
answer
can
is
have
still
2
yes.
genes on gal4, right?
Yeah,
You can't.
yeah. No,
Oh, you express
can-
both of them that way.
I'm not supposed to talk.
I just
Okay, fine.
just out of curiosity.
I'll explain it later to you.
Okay.
Out of curiosity, how long would it take? So you guys are gonna make the rescues, right? So how long till? Do you know how long it would take to make those?
To cross them, cross the flies themselves.
And you might not. I'm just curiosity.
I think we
Takes
said like a
2 weeks to breed them.
couple weeks. Maybe I think we can.
Oh, yeah.
Yeah.
All right, that's awesome. Okay, just thank you.
Wait, I didn't finish.
Oh, wait, she didn't finish, just one.
We are hoping for 1 or 2 temps who have about the same training in flies as Sophia and I.
Just none.
Yeah. And hopefully they can continue the project with Sophia next year. And we both are going to do sports in later trimesters, but not right now. So we're going to work after school, after sports, during free CPs and iRT CP. Our proposed budget for this year is about $950. So our fly strains, which is going to be a lot of strains and a lot of flies, should cost about $50 to $100, and they will last past this year, since I believe Dr. Kirkhart's going to take them home. Yeah, for the summer. Our fly husbandry stuff, which is the food, vials, anything that we need to in order to make them live, is going to be about $300.
I'm good on costs. Are you guys good on costs?
Yeah.
I think we're okay. Thank you.
Sounds fine.
Okay.
Yeah, so
Thank you.
that's.
Woo!